Hi, and welcome to PubMed for Scientists. Today I'm going to be covering a lot of ground in a short amount of time for a broad audience. So after an introduction to PubMed, I'll talk about searching, narrowing your results, saving and sharing your results, setting up email alerts, and finding related information. And throughout this talk, I'll be touching on the advantages of logging into your MyNCBI account when you use PubMed. And just to let you know, I'm going to start out with some slides, and then I'll go into a browser to run through some searches, and then I'll finish up back in PowerPoint. There are currently more than 25 million citations to published biomedical literature in PubMed, and most of the citations are for English language articles, and they include abstracts. As I've said, this is a citation database. About 20% of the citations in PubMed link to free full text, either in PMC or at the publisher's website. And if you look at the last 5 years, the percentage of citations that link to free full text jumps to about 35%. This is due to author manuscripts being deposited in PMC and cited in PubMed, and an industry increase in open access. So let's talk for a minute about PMC or PubMed Central. Much of the free full text that's available is available because of the NIH public access policy. This policy requires that published results of research supported by NIH must be available to the public. Final peer-reviewed manuscripts accepted for publication are available to you through PubMed Central, and they are cited in PubMed when published. PMC is a separate full text repository for these articles. There are also links to full text if the full text is available from the publisher or through vendors like EBSCO, ProQuest, Highwire, Ovid, or more. The publisher will decide pricing and availability, so the other full text is not necessarily free full text. But often students, researchers, medical professionals will have access to full text through their institution's subscriptions. So please talk to the librarian at your institution who's responsible for your online subscriptions to find out how to access them. If you're at an institution that has a medical library, please look into the library's holdings, log into PubMed through your institution's specialized URL, and take advantage of the journal literature available to you that your institution is already paying for. Okay, before I get into a search, I'm going to go ahead when I'm live in PubMed and make my screen a little bit bigger. So I wanted to just go ahead in PowerPoint and look at a snapshot in PubMed before we get started so you can see the whole screen in one shot. So the PubMed results screen can be a little bit overwhelming, and some might say it's busy, but I like to say that it's packed with tools and information that enrich your PubMed experience. And I'm quickly going to go over the various sections of the screen before we get started searching, mostly so that you'll know what words I use to refer to the different areas, so that when I get into searching and I'm talking to you, you'll know what I'm talking about. Of course, we're all familiar with the search box. And the PubMed label is also a pull-down menu, and when you click on that arrow and pull down the menu, you can access other NLM databases, things from gene and nucleotide to the NLM catalog and PMC. Your search results appear in the middle of the screen. I think we're all familiar with this area. We have the left sidebar, which is the filters sidebar menu. There are actually menu options across the top above your search results where you can customize your display and also send your results and share them. And we'll talk a little bit more about those menus. On the right, we have the right sidebar, which contains more information for you and links to additional resources. And the right sidebar menu will change depending on your view. When you're at the results screen and you're looking at all of your results, you'll see certain options. When you're looking at an individual citation, then the citation level information might be a little bit different. You'll see at the top of my screen that my email address appears because I have a MyNCBI account and I'm already logged in. If you don't have an account, you can register very easily. I won't go through those steps here, but I did want you to know that my screen will look a little different because I'm logged into MyNCBI. And I mentioned the NIH public access policy. MyNCBI's My Bibliography feature was designed to help manage authors' manuscripts. And many of the features that I will point out for saving and sharing your searches, customizing the way your results will display, and saving your search history will come from MyNCBI. And then there's just a few other links. Some people don't realize these are links if you want to get to the PubMed homepage or NLM or NIH. You can also get to the NCBI homepage very quickly. And of course, there's online help available for you. Okay, before I get into PubMed, I just want to quickly talk about the best practices for searching. When you search PubMed, you want to be specific. Type in all of the terms that you need to use to describe your concept or problem or question. Don't use punctuation, no quotes, no asterisks, and you don't need to use operators. PubMed is very good at breaking your search into concepts and adding the Boolean for you. Simply type your search terms into the search box and hit enter. So I'm going to go ahead and switch to my browser now. So I talked about access to the full text from the publisher or aggregator. You might also have access to your own institution's subscriptions. And we have a free service called Link Out for Libraries, where your library sets up an account to share their collection via PubMed to authenticated users. Links that are set up in PubMed appear as icons that you can click on for either immediate access to full text or a method for ordering the article. If your library participates in this program, you'll see your library's icon on some or all PubMed citations depending on their service. So I'm going to go ahead and show you very quickly what that might look like at your institution. I went out to the UIC library, and from a library homepage at a university, for example, there typically is a selection where you can get to databases. This happens to say databases A to Z. And then, and again, this is pretty typical. I can scroll down to PubMed, and if I were at UIC, I would click on this link to access PubMed. And you can see the interface doesn't change. It looks the same, but when we get into our searches, you'll see that I've activated that UIC icon. On my citations, I don't have the privileges to go through that link and order the materials, but you can see what that icon looks like on the citations. Okay, I'm going to go ahead and start searching PubMed. And the first search I'm going to do is 3D printing. And you can see the autocomplete feature here. I'm going to go ahead and use that autocomplete to select 3D printing tissue. Now you can see here I've got a retrieval of more than 508 citations, and that actually has generated this results by year chart on the right sidebar. This graph is there to show trends in publishing, and you can certainly tell when you look at this results by year graph that this is a very current topic. And you could download those PMIDs, the number of PMIDs by year. You can download if you click on Download CSV. Now to see how PubMed processed this search, I'm going to go into the weeds for just a second here and show you that translation. If you scroll further down on that right sidebar on the page, you'll see the search details section of the screen. If you click on See More, you can see that in its own window, and so it's a little bit clearer. And you can see here that PubMed has translated this search very well. It's going to process the search, mapping to both 3D printing, which is the way that you typed it, and also printing three-dimensional. So what you're looking at in this expanded view is that PubMed is actually finding the medical subject headings, which is the specialized vocabulary describing concepts in literature. And MESH improves your search by mapping synonyms and accounting for variation in language. And the reason I'm showing you this is why we don't want you putting your search terms in quotes, because it's this translation that you're turning off. When you put your search in quotes, you are making your search literal and turning off this mapping that PubMed does with with your terms to make your search more comprehensive. So for this search, I'm going to assume that I'm interested in a general, quick getting up to speed on this topic, and I can use the left sidebar menu to limit my results to review. And you can see when I do this, as I scroll down my results, it's pretty clear that these are all review articles. I mentioned that most citations contain abstracts, and it's important to note that often we find our users are able to get much of the information that they need from the abstract without having to pursue the full text. And I'm going to show you the display menus quickly. The default display is summary, but if I want to be able to scroll through my results and get a little bit more information, I would switch to the abstract display. And then as I'm going through right there, I can see the abstract, and then I can see also some of those links to the full text. Now, if I'm at my results list and I want to look at some of these citations and read the abstracts and link out to the full text maybe at a later time, I can actually select the citations that I'm interested in. And use the Send To menu to email those citations to myself. I could also email them to a colleague if I wanted to. So I simply click on email. Before I do that, I'll let you look at the other options here. You can send them to a file, to an NCBI collection, a citation manager, and then we'll look at the MyBib feature in just a few minutes. So I'm going to go ahead and select those 4 items. You can see that my email address is already pre-populated, and that's because I'm signed into my NCBI. I could write a note to myself. If I was sending these to a colleague, I probably would write a little note just so that they'd know the context that I'm sending them from. If I click on email, I'll see those in my mailbox probably within 2 to 5 minutes. Okay. One of the most popular searches that's done in PubMed, and one of the searches we get a lot of questions about, is the author search. So I did want to take a minute to do an author search and talk a little bit about author disambiguation. I think author disambiguation is an issue in publishing in general, and PubMed has a few ways that they're working to help identify specific authors when you search for an author in PubMed. So first, I'd like to point out that the proper syntax for searching for authors is last name space initials. And today I'm going to search for Dr. Murray. Oh, I'm going to clear my filters. So I've searched for Dr. C.J. Murray, and I know Dr. Murray does his work in the area of global burden of disease. If I were getting a lot of results that were not related to my author, I might add a subject term to this search to make it more specific, but I feel like I'm getting a really good retrieval. And I know, for example, that this second citation is definitely the author I'm looking for and the topic that I'm looking for. I'm going to change my display really quickly to that abstract display so that I can see the affiliation information. And in the past few years, PubMed has accepted and displayed more affiliation information. It used to be limited to the first author. Now we have the affiliation information for all authors for citations submitted more recently. And so I can look through this affiliation information, and since I can actually see that my author is affiliated with the University of Washington, Seattle, I know that that's the author that I'm looking for. If I click on his name, PubMed is now doing a sort by author sort. It's a computed sort by author that takes into account co-authors, topics, affiliation information, subject terms. And now I'm pretty confident that all of Dr. Murray's citations are going to be in this first or second page, depending on how many citations he has in the database. If I actually was Dr. Murray, I could select the citations that were mine and create a bibliography, and I'll go ahead and show you quickly how you would do that. I'm going to just go ahead and grab 10 of these citations, although I know there are many, many more. And I'm going to send them to my bibliography using the Send To menu. I'm over in the My NCBI set of features in my NCBI account, and I can click on Save, and it tells me that it has added the items to my bibliography. I want to go back in my NCBI and edit this bibliography. Now you can make your bibliography public or private, and it will tell you that you've updated your permissions successfully. I'm going to make this bibliography public, and I want to show you that I can grab this URL for my bib, and I can share this now either through social media or email. I could embed it on a web page, and if I were to email that to someone and they clicked on that link, you can see that they get a nice clean list of my publications, if in fact I were Dr. Murray. All right, I'm going back to my author results, and I just want to talk for a couple minutes more about other tools for author disambiguation. I think it's important to note that in the past couple of years, we get questions about whether or not NLM would ever come up with some kind of identifier for authors, and that's really outside the scope of anything we would do. And it's already existing in industry, and there are institutions or groups that are issuing unique identifiers for authors. And we're seeing the most popular ID is from ORCID. It takes about 30 seconds to register at ORCID and get a free author identifier, and we accept these identifiers when they're submitted with the literature. We are not retroactively able to apply them to citations, but if you submit a unique author identifier with your work when you submit it to PubMed, that will be attached to your citation, and that is something that's becoming more and more prolific in the database, and it's really going to be the key to being able to search quickly and retrieve your citations in the database. Okay, I'm going to switch gears again and get back to some subject searching. And you have to bear with me for a second here. I'm going to search for microRNA expression lung adenocarcinoma. And now with this search, I'm going to go ahead and use a couple of other filters from my sidebar filter menu. I want to point out that there is a free full text filter, which is very popular. So I can restrict my results to citations that are available, that the full text is available for free. And then I'm going to go ahead and customize the publication dates for my search. I'm going to set up a custom range, and what I want is all of the citations from 2015 forward. And when I put in the 2015 as a starting date, you can see that my filter now says from 2015 0101. And if you look at that again, you can see that it has defaulted to a date way in the future. So I'm grabbing everything that's in the database from January 1st, 2015 and forward into 2016, because we do have some ahead of print citations in the database already. If I wanted that to limit that to 2015, I could change that end date. And you can see that PubMed knows to default to the end of the month. And it will change the label on that custom date, so I can tell that I've restricted my results to publications within this year. I'm going to go ahead and change my sort order to a relevance sort. And if you look at that sort menu, really the two most popular are right there at the top. It's a relevance sort, and then also we have a sort by most recent, which is the citations that were most recently added to the database. These two are really the most popular, and we do add citations 7 days a week, so that most recent is used quite a bit. But I'm going to switch to a relevance sort, and then I'm going to look at this first citation that I've retrieved. And as I said, when I click on this citation, and I'm now looking at the citation level information, that right sidebar menu changes a little bit, and I'm getting citation level related information. So you can see here I've got similar articles being recommended to me. I can see what articles in PubMed Central have cited this one. And then I get down here to a related information section of the screen, and I can see here the articles that are frequently viewed together. So this is more of a user behavior type of link where people who are viewing this main article that I'm viewing have also looked at other articles, and I can show you that very quickly. I know that the 3 articles I retrieve I won't see as long as my filters are on, but that will give me those citations that people are looking at when they look at my citation. And then I can use this recent activity section to go right back to my citation. You can see the link there to PMC as well. Now, before I go any further in the related information section, I just want to talk a little bit about the data that is available, the molecular data that's available at NCBI, and provide a little bit of context. So the basic challenge of genomic data is that genome assembly really can be problematic because genomes themselves are quite large and contain long stretches of contiguous sequence, really on the order of millions of pairs, of base pairs. And the commonly used genome sequencers can only generate relatively short segments of sequence. So a genome must be fragmented, sequenced in bits, and then reassembled to obtain the full sequence. So just before I get into the weeds here with some of these databases, each sequenced piece of DNA is referred to as a sequencing read, or a read for short, and both raw reads and assembled data and maps to assembled data are typically available through the various NCBI databases. And the takeaway here really is that these data are available for you to search through, download very quickly, and use for your own analyses. So let's look at this related information. There are 2 different ways that the associated data gets linked here. Traditionally, the link has been to data reported in the article, and in this case, there are links to the reported gene expression series in geodatasets. And if you hover over these resources, you'll get a little synopsis of what kind of data you'll find in there. And the SRA data also is referenced from the article. In most cases, the related project link, which leads to the NCBI bioproject database, is the easiest way to access all of this directly associated data. So you can see here on this screen that the bioproject record has links to the GeoSeries and the 12 supporting RNA-seq experiments. Details about the individual samples are available in the biosample database. So I'm going to look at this list of SRA experiments. Now, these data comprise RNA-seq for small RNA and total RNA of tumor and normal lung. And note that you could even perform your own analyses of these data using BLAST at the NCBI site. I'm going to go ahead and select one of these. And then I could use the Send To menu to send this to BLAST. There we go. And now we're in BLAST. And while I'm here, I will just mention that I know NCBI has other webinars and NCBI minutes that talk about how to use BLAST. And actually, a lot of the NCBI databases, there are learning resources available. I'm going to go ahead and close out of here and go back here. Okay, again, I'm going to use this recent activity to get back to my citation. Now, the other type of link available through the related information section, and I'll scroll down there again so you can see it on the screen, is to molecular information brokered through other NCBI resources. So the various links to gene, to nucleotide, protein, unigene, and geoexpression profiles are created through the inclusion of the PubMed citation on gene records, either directly through citations on sequence records or through gene reference into function or gene riff submission. And you can see there is a lot of data here. The links to MedGen, which is NCBI's medical phenotype database, are made through mesh terms on the PubMed citation. The links to references are provided through the full text version of the article, will provide the full text version of the article in PMC. And if you click on gene, you can quickly get access to the gene records that cite this current article. All right. Very quickly, I'm going to show you how to use the advanced search interface to add terms that will find particular types of linked data in PubMed. So if you go to the advanced search screen, you can use a filter, for example, to limit your search results to citations that have gene assembly data available. And this is sort of a very much in the weeds thing, but I know it's a tool that scientists actually really like to use to get right to the citations that will have the data that they can use and analyze. So you can see if you select that filter field from the search builder, type in PubMed, you'll have a list of some of these databases that we've looked at, and you can select assembly, for example, search, and now you know that all of the resulting citations will have assembly data that you can look at. And I'll just click on one of these very quickly and go to assembly. And you can see the assembly sequence data that's available. And you could narrow your search if you wanted to when you're back at that screen where you have that filter search. For example, I could add a topic term like plant pathology. And again, I don't need to put in the Boolean or any quotes or anything fancy. And now I've limited my results not only to citations that link to assembly data, but also I've now limited to a certain topic term. Okay, I have one more quick search, and then we will open up for questions. I haven't been monitoring the chat box, and I'm very curious to see what's been coming in. So when I conduct this search, I just want to show you very quickly a couple of filters that you could apply. For example, again, if you were interested only in reviews, you could limit this to review, and that would limit your results quite a bit. If this is a topic that you're interested in on an ongoing basis, you could also save this search and create an alert for it. And you could change the name of the search if you'd like. You could modify the search. You don't want to modify the search terms. You want to definitely modify the title, not the search terms. You could test it again, but since we just ran it, I'm going to assume it's running just fine. I would like to receive email updates of new search results. It has auto-populated my email address because I'm in my NCBI, and I think I'm going to switch this schedule to weekly and look for my results on Monday. Summary report is just fine. 5 items at most will be sent in the email. However, the email will also contain a link, and when I click on that link, I will get the full results of my search. And I always recommend checking this box so that you will receive an email even when there aren't any new results. Otherwise, when the email doesn't come, I think people tend to be a little afraid that they missed something. So this way you'll get an email regardless, and it will tell you when there are no results. I can save that search. And then weekly, I will get updates and I will see the new citations that I haven't seen before related to this topic. One last thing before I wrap up here. If I go into my NCBI, I can show you there are a lot, 4 or 5 different windows I think that I have open. You can customize this display for yourself. You can see my bibliography that I created earlier. I have a lot of collections that I've created. But what I want to show you is this recent activity, because when you're signed into my NCBI, PubMed will keep your activity for up to 6 months. And it also provides you a nice one-stop shop for all of the databases that you were in. And you can see here that we've been in PubMed, Assembly, Gene, SRA, Bioproject. And all of these searches are here. I don't really have to think about saving my searches or writing things down. I know that I've got this history, and I can quickly repeat these searches. I'm going to go ahead and wrap up my part of this presentation. I have a slide. This will be made available later today. And on the first slide, I'm going to go ahead and put this slide up because this will get you to all of the materials. You can see that URL on this slide that will link you to the materials. And we'll have the presentation, my notes, the questions that we answered today. And here I've got links to the webinars and NCBI minute recordings on more topics. And then also at other NLM online training resources. For example, for the PubMed, there's a PubMed tutorial and there are quick tours on lots of different searching in PubMed, including author searching, searching for drugs and chemicals. So I encourage you to create a MyNCBI account, register for an ORCID unique identifier, talk to your librarian about your resources, and then link out to some of these tools that we've got online for you.
Okay, one of the questions, actually a couple of questions about ORCID. Starting in January of 2013, NLM began accepting from publishers author identifiers when it's included with the work. So there are a few different author identifiers out there. There's ORCID, there's International Standard Name Identifier, there's Virtual International Authority File. We accept all of these, but again, they need to come from your publisher. So when you submit your works, do include your ID. Now, as Marie said, it just takes a minute to get one of these. So you can take a look at the orcid.org website. And when that is included with the article information that we get from your publisher, it is there in the file. So hopefully you're seeing the demo on your screen where you can see that identifier. We name the source, and then there's the number. So that once this information is included in the PubMed record, it is searchable. So we don't have a huge number yet, but as the years go on and more authors do this, it's going to be a really useful tool. So please do so. Okay, another question that came in, could you filter by study type? So I'll have Marie show you the different article types available in the filters menu. You do need to expand that by clicking on customize, and you can see that we have a wide variety of publication types and research types available in this menu. Yeah, go ahead.
I don't know if you can hear me, but since the question came up, I would like to say customizing the menu is sort of a two-step process. So when you, let me go back in case you missed that. If you click on customize, then all of those article types are available to you, and you can select the ones that you want to add. If I want to add comparative study, for example, I would select it from the list and then click on Show. Now that menu appears on the left sidebar filter menu, but to activate it, I need to click that selection. So it's not activated until you go ahead and select it. And I think sometimes some people overlook that last step.
Let's see if I can find it again. There was a general question about mesh and the structure of mesh. Here we go. Are mesh terms highly hierarchical, such that there are 5 or 10 mesh headings that each pull up 10 or 20% of all PubMed citations? Well, mesh is in a hierarchy, and actually if you use your database selection menu up at the top of PubMed, you can actually go into the mesh database and explore the vocabulary if you'd like to. And at the bottom of every mesh record, it shows you the position of the term in its hierarchy. And yes, some terms are very broad. However, we like to say, please be as specific as possible when searching PubMed. It's a huge database. So if you can try to find the term that most specifically describes the topic you're interested in, you're going to end up with much better results than if you put in general terms. There was a related question. Oh, well, the related question was actually a specific research question, and that was how to search for congenital heart defects in Native Americans. And I think if you put that right into the search box, congenital heart defects, I'll let Marie do it. Native Americans, yeah. If you run that search, you might want to clear your filters and take a look at your search details. You'll see that you have mapped specifically to two mesh terms that describe your two concepts. So congenital heart defects gets mapped to heart defects congenital, and Native Americans gets mapped to Indians North American. So in our estimation, that's actually a pretty good search, but you only have 21 results. So this is I think a rather unusual case where you might actually want to do a little bit of a broader search. And you could check mesh under, for example, congenital heart defects, or it'll be in the mesh database as heart defects congenital. And you might want to bump that up to the next level. I think there's a broader term for heart defects that you could try to see if you can find some more research in that area. So here you see the mesh terms. There's heart defects congenital, and if you click on the term and jump over to the mesh database, you can get a description of the term. You can, at the very bottom of the record, see the position in the hierarchy. So there are quite a few specific congenital heart defects, but you can also go up one level to cardiovascular abnormalities. So you might try that. So hopefully that helps. Or even as high as cardiovascular diseases, yeah. There was a question about systematic reviews. I'd like to address that one, if I may. Not to monopolize here. Systematic reviews are increasingly popular, a very useful tool to search to find out the best information about a particular research question. Unfortunately, in the mesh vocabulary, we don't have a specific publication type for systematic reviews. So what we've done here at NLM is we've developed a search strategy that allows you to find systematic reviews, and other information we think would be useful to answer a clinical question. And you can access the filter, which enables that search strategy. In that filters menu, again, going back to the article types, you can find systematic reviews there. So if you activate that filter, it's activating a search strategy, a strategy it's running along with whatever you put into the search box. And in your results, you will find systematic reviews. You will find other stuff that's hopefully useful, but you will find systematic reviews that way.
Okay, there were a couple of questions about some of the things, the behind-the-scenes stuff that PubMed is doing. One of them was about the relevance sort. How is that relevance determined, essentially, I think is the question. And also, a person wanted to know more about the computed sort by author. Basically, how does that work exactly?
Hi, this is Kathy. So let me chat a little bit about relevance search first. I've opened the PubMed help window, and if you scroll down to the appendices here, you'll see we have a computation of weighted relevance order in PubMed. So this is the actual algorithm in as few words as possible, explaining how we generate relevance based on global weight of term, local weight of the term. This algorithm was built by the same group that brought similar articles in PubMed. So the information is there. And I believe there is not yet a link to the published article because it is not yet published, but the authors of this feature will publish this in the literature soon. So as soon as that link is available, I'll add it to the help. The other question was about clustering of authors or author sort. So what we do there, again, that publication is in the works, so there's not something in the literature just yet to link to. But as Marie pointed out, when you run a search in PubMed, if you click on any of the authors that are associated or listed in a PubMed citation, you'll get a link to those authors or the authors with the same name that are published with the similar author list, that have the same subject, affiliation information. So here we've got Cutting GR as the author, and there are 191. Chances are that there are more than just one GR Cutting in the database. So what we do is rank the information so that those citations that we are fairly confident were written by the same GR Cutting that we clicked on from that original citation are listed first. And we found that when users run just a general author search versus running the author search from the abstract, they more often click on the first few citations from the clustered or sorted by author listing. So we're fairly confident that we're finding the correct author in the first couple of pages. I hope that answered the question in a roundabout way.
This is Peter Cooper. There was a question about the links to data, which was one aspect of what Marie was talking about today. And the question was, are those links maintained? And the ones that, as Marie talked about, there are basically two kinds of links. The ones where the data was reported essentially in the article, I think those links are essentially permanent. And those, essentially they're an archival link and they will always be there. The other ones, the ones that are the sort of the value-added links, like the links to gene, those are maintained. One of the aspects of the question was sometimes gene names change or other aspects of the gene record changes. We're doing that. So the citations that were put there on the gene record will follow the gene through any updates in the gene record. There was also a question about, and I think I sort of misled some people when what I wrote back in the chat box. One of the things that can be a little frustrating is when you're trying to select individual records, say in a PubMed retrieval, so like you have to check the boxes. There isn't a way to just to select all 20 on a page or whatever, but you can, for example, download all of them if you want to. If you go to the Send To menu and you download and you go to Send To, say you send a file, for example, you're going to get all of them, not just the first 20. So that's one way to get everything there if you need to do that.
I had a couple of basic questions about mesh, what it is, and a couple of folks who maybe are complaining a little bit about mesh. So let me just step back a minute and explain. MESH stands for Medical Subject Headings. It's our vocabulary for describing the literature. So it's a controlled vocabulary. So it's a set list of terms that indexers and catalogers choose from when they try to describe what an article is about. So those MESH terms get placed on a PubMed citation so that you can then use that controlled vocabulary to search with, but more often it's PubMed mapping whatever you're typing into the search box to MESH to enlist the assistance of that controlled vocabulary to get better results. So the bottom line really is, again, try to be as specific as possible when you search PubMed. Check your search details and make sure that you are mapping to MESH for any medical term. MESH won't handle other types of terms very well, but if for any medical term, you should find a map to MESH. And if you don't, try a synonym. And if there is a case, I see one example here, where you think that MESH just isn't it's failing for you. You have a good medical concept, but there isn't a MESH term for it. Please write to us. On the MESH database page, you can find a link to the MESH homepage, and we love to get MESH vocabulary suggestions so that we can improve that vocabulary. It is a hand-curated by human beings vocabulary. So it is updated once per year, but we work on those terms throughout the year.
I think we need to go ahead and wrap this up. So there are more questions in the questions pod than we were able to answer in the amount of time, and that's good actually, but we will answer them. So we'll get those in a document, and the ones that we talked about we'll also answer in that document. So all that stuff's going to be available linked to that URL that I gave at the beginning. And once this webinar is recorded and on YouTube, I'll also send out an email to everybody that signed up that it's available. So let's just show the last slide that has the links to things. Okay, so just a reminder that we do have the webinars page, webinars and courses page, and so that's where this stuff is going to be linked. And as I said, I will send out an email to everybody that signed up. Thanks everybody for coming, and we'll talk to you next time.